It's a great pleasure to be here at the Pandemic Innovative Conference. It's a really wonderful gathering for this important problem, and really grateful to the organizers for inviting me. So what I'd like to do today is tell you a little bit about the race for the vaccine that's currently underway globally. And it's really impressive to see the amount of innovation that's taking place. This is a plague that took place a few hundred years ago, 1665, the Great Plague of London, killed about 25% of London. And this gentleman, Sir Isaac Newton, was forced to move from his home institution of Cambridge to his family homestead about 60 miles north of London, and use that time and that situation to be really innovative in a whole bunch of different areas: the creation of calculus, development of optics, the early theory of gravity. And I think we're seeing something very similar in our own past couple of months, is the level of innovation. I've sat at MIT for over 2 decades, and the amount of innovation in a compressed time is really impressive. This is not just in the area of COVID itself, but in many different areas of science. This is a new, I'm a physicist by training. This is a new approach to physics that I'm personally really excited about. Lots and lots of innovation. So what I want to do today is tell you about some of the exciting approaches that are being taken towards the SARS-CoV-2 virus. And as mentioned, it's really the number of different types of approaches that is most striking. These are a set of micrographs that come out of Elizabeth Fisher's lab at the National Institute for Infectious Disease. Really beautiful electron micrographs showing the CoV-2 virus, coronavirus. And you can see on the surface these little nodules. These are now famous so-called spike proteins that allow the virus to interface with human cells attached to human cell and inject their genetic material. And if you think about what a virus is, a virus really consists of 2 parts. It's the genetic code, or the genome of the virus, that codes for the virus itself. So every living thing has a set of nucleotide bases, A, G, T's, and C's. We have our own human genome, which is about 3 billion bases long. This viral genome is about 30,000 bases long, 1/100,000th the size, but still pretty sophisticated. So this piece of RNA, in the case of the coronavirus, codes for all of the structure of the virus that you see here, including the spike proteins that interface to human cells. So this is just a snapshot of what that genome looks like. The parts of the genome that code for the proteins of the genome, and various pieces of machinery that allow this virus to replicate. And so it's instructive to take a look and see what processes the virus goes through to replicate itself. So this here is a schematic of the virus, the genetic material inside, the spike proteins on the outside. This large area here is the human cell. And these spike proteins interface to a natively resident receptor on our human cells called the ACE2 receptor. These spike proteins fit perfectly into this ACE2 receptor. They allow the virus to attach to the surface, and once a viral particle is attached to the surface, it gets endocytosed, sucked inside of the cell. Once it's inside the cell, the viral package opens up, exposing its own genetic material, which is then translated into a set of proteins that help process this genetic material, ultimately ending up in the replication of the virus. And so just seeing both the electron micrographs that I showed and this schematic picture, right away we can start to think about strategies of how to combat or prevent this viral infection. And the most natural or most obvious thing that one can see right away is, if I could cover these spike proteins, if I could figure out a way to cap them so they don't interface with my own receptor, my ACE2 receptor, I'd have a way of preventing this infection. In fact, this is what the immune system does. The immune system creates antibodies, which are themselves other proteins that can be programmed to bind to very specifically to viral proteins, for instance. And so what the body wants to do is create antibodies that can bind to these spike proteins to prevent infection. And the way that the immune system works is that if you present a foreign protein, such as the spike protein, into the body, the immune response kicks off a series of events that results in the creation of antibodies that will bind to these proteins. And so much of the effort and goal is different ways of presenting the human body with these spike proteins. So I just want to remark that while in the past, the way that we've done that is to create or take deadened virus or attenuated virus and re-inject that into the body to attempt to elicit that immune response. This harkens back to the original vaccines of Jenner back at the similar time of Newton, by the way. Those all relied on the genetic materials or the proteins that existed that were made by that infectious virus itself, what nature gave us. We now have a pretty impressive set of advanced tools that allow us to go way beyond that. And they allow us to both design novel proteins, so-called computationally designed proteins, and then synthesize the DNA that codes for those novel proteins. So these are proteins that never existed in nature. They've been designed on a computer, and they may be designed to look similar to the spike protein, but have various enhancements. For instance, they might be enhanced to be more stable so that they create a greater immunogenic response. Or they may be enhanced so that they are expressed and produced, manufactured at higher levels. And so now what I can do is I can come in, this is some work actually that came out of our lab a number of years ago, so-called next generation chip-based DNA synthesis, where I can take computationally designed proteins, I can take the DNA code for those and synthesize those on a specialized silicon chip, and now create so-called synthetic biological vaccines. And the third ingredient here, or the third tool, are a set of tools which are even more sophisticated than that. These are tools that allow us to go beyond the human immune system, go beyond antibodies and target viral DNA or other DNA sequences directly, so-called CRISPR tools. And this is an area that our lab has worked on considerably as well. So I want to give a quick overview in the few minutes that we have of some of the significant and major efforts that are afoot in the pursuit of vaccine and therapeutics targeted at COVID-2. Up in this chart on the upper two left are the traditional vaccines. These are taking the virus itself and either killing it or attenuating it and re-injecting it to induce the immune response. But the major efforts that are at play right now are really dependent on the tool set that I mentioned, so-called synthetic biology tool set, where I'm come in with synthetic DNA, and I use that to express either parts of the virus, the spike protein, or reprogram another virus entirely, a virus that may be benign in general to us, but has now been reprogrammed to express the COVID-2 spike proteins on it. This is the approach up in the upper right that's being taken by companies like AstraZeneca and J&J. These are already in advanced trials. The AstraZeneca has been supported by BARDA, which is a U.S. part of the health service. More than $1 billion has been put into this, and phase 3 trials already started with a goal of about 10,000 participants in that trial. Really unprecedented numbers in terms of the number of volunteers for these trials. One of the most exciting vaccines, I think, is this one in the bottom right, so-called DNA vaccine. And here, this DNA vaccine actually harnesses the human body itself manufacture the spike protein. So in the upper right, you're actually coming in with a virus that already has the spike protein. Here, you're just injecting the DNA code, in the case of Moderna, an RNA code, which instructs the human body itself, just like a virus, to make this spike protein. That spike protein is then expressed on the surface of the human cell and elicits the immune response. This is a vaccine that has not been tried before, but really exciting approach in its kind of nimbleness for being able to change the code as the virus mutates. We need to be able to keep up with that and to be able to change the vaccine itself. If you look at what is rolling out in terms of our capacity, one of the reasons so much resource has been put at this is many of these are being scaled in production and manufacturing prior to the completion of these phase 3 clinical trials. So this is, again, pretty unprecedented. The goal across the board here looks like it's on target or hope to be on target to produce somewhere around 3 billion doses globally by the end of 2021. Really an unprecedented number, but if you think about our global population, more than twice that, we're still a factor of 2 away from where we want to be. And so I think one of the exciting approaches or important approaches is not injected vaccines, but so-called oral vaccines. These are vaccines that are taken orally. And what's striking or useful about this is that these can be scaled up in fermenters. These are things that look like the machinery that makes beer, for instance, or many of the flavoring components that we have globally. And the amount of global capacity to scale here is considerable. This should scale to many tens of billions of doses annually. And this is really an important direction. One of the significant players in this is a company here in Israel called Migvax out of the Migal Institute. And this is a really important direction. Okay, so what can go wrong? Many people have asked, aren't there many viruses that we have not been successful at making vaccine to? And the answer to that is yes. We've got viruses like HIV that we've been unsuccessful at. And but if you think about that, a number of those, for instance, HIV is an integrating virus. As I mentioned, it integrates into our own genome, makes it a very difficult problem, although CRISPR may be successful at that. We now have the tools, I think, even to go after very difficult viruses like that. Another problem in the past has been so-called ADE, antibody-dependent enhancement, where you have kind of a weak vaccine that kind of loosely binds the virus, and that, ironically, increases infectivity. It draws the virus towards the cell without neutralizing it, and actually increases the infectivity. I think given the tool set that we have and our ability to design antigens and antibodies in a way that we have not previously, I think the evidence is that we will not have these issues in the current virus and are actually on a good path to solving this problem. Just really quickly, there is a whole different path, which are called therapeutics. So these are for folks that either have COVID already, so creating a vaccine is too late for them, or a prophylactic therapeutic, which you could have systemically in your body that would prevent or destroy the virus in a totally different means other than the immune system. This is a paper that came out of our group. This is Pranam Chatterjee and Manu Panapati. These are computationally designed peptides that bind to the spike protein. They then are tagged and tag the virus with a so-called ubiquitin tag that kicks off a set of machinery inside the cell, so-called proteasome, that digests the virus. And we have some really exciting preliminary data that's based on this approach. This is really a new approach to a therapeutic. And then this is also an effort that came out of MIT. This is from Kfir Schreiber and Frost Karidis in our group at MIT that have spun off a really exciting company called DeepCure. This is a machine learning platform which can look at a very large part of chemical space and look for therapeutics, small molecule therapeutics that can go after different parts of the viral replication cycle, so-called protease. And what's exciting about these approaches is that they have the prospect potentially to be broad antivirals. So not just against COVID-2, but to be able to go broadly after many, many different viruses that all rely on various proteases. So I think the exciting kind of directions here is that there's many different pieces of innovation that are coming together. I believe these will serve us well, both for COVID-2, but also set us up well for future pandemics, future threats that we have in this arena. So with that, I'm happy to take any questions.
Wonderful, thank you. One question, does COVID-19 appear to be lab-made or naturally occurring and make a difference in terms of finding a vaccine?
Yeah, my strong my strong hunch is that it is natural. It's not lab made. I don't think it makes a difference. I think we've got a sufficient tool set, both for if you look at the rapidity, the rapidness with which we both sequence the virus, create a crystal structure to the virus, and kind of deduced its infectious path, and then the set of other tools that we can bring to bear on the solution. I don't think it matters in this particular case. This is not, as I mentioned, the most difficult of classes of viruses. There are more difficult classes, but I think part of the one thing we should, I think, be grateful for is that this has really harnessed a huge amount of resource and kind of global energy in this direction that I think will set us up well to combat future threats that come our way. Hopefully not man-made, but even if they are, I think we are in a vastly better position than we were just a few months ago.
Okay, and another question is that knowing that there's no vaccine currently for SARS, how are we confident that there will be for COVID?
Yeah, well, I think SARS, I think there would have been a vaccine for SARS. SARS was a problem that kind of went away on its own. It didn't materialize into the kind of pandemic that we have right now. And so I think if that had continued, we would have a vaccine for that. I don't think there was a structural reason that we don't have a vaccine, other than just market forces didn't make it worthwhile to continue that.
Okay. Any further questions, or would you like to wrap? Oh, here's one. Given the 80+ efforts around the world, does MIGVAX have a realistic shot at being an early solution?
Yeah, well, as I mentioned, I think I would advise you to speak to the folks that are involved in that company if that's something that you are thinking about getting involved in. What I am excited about in regards to MIGVAX is just what I mentioned, is the ability to scale the manufacturing of that, I think, to many tens of billions of doses, I think is a really important attribute of that effort.
Wonderful. The closing plenary will begin in 5 minutes. Joseph, do you want to say any last words here?
Just that really excited that the number of participants and the amount of kind of global interest in this issue, and also that I think I and many of my colleagues are enthusiastic about the amount of innovation that's come together on this. I think we're enthusiastic about the prospects of meeting this challenge and that in turn setting us up well in preparedness for next challenges that may and will come.